right, we've got about 27 people here. Welcome everyone. Can everybody see my screen okay?
Yes.
All right, good deal. I will give people a couple of more seconds to pour in. Nice crowd. This is kind of a who's who of the misophonia community.
Is this session meant for sufferers or for scientists, researchers?
Anybody who wants to attend. So yes and yes.
Thank you.
Sure, good question.
Mm-hmm.
All right. Okay, I'll go ahead and get started. There's a few people coming in. Michelle can let them in. And I've got a few things. Oh yeah, Heather says while you're waiting, if you're comfortable, go to slido.com and look up FOAMS, foams, and share your misophonia triggers. There's research happening in real time while we're doing this. It's amazing.
That's right.
Okay, I'm going to go through, I'm Chris Edwards. I'm the founder of soQuiet, and I'm going to go through some, they call it housekeeping, but you know, it's the etiquette and a few requests for these types of sessions. So feel free to come in. This is a soQuiet science session, and here's a few things to go over. First of all, if you're an attendee, this is technically a meeting, but we kind of run it like a webinar. We're kind of cheating the rules of what Google Meet can do. So if you don't mind keeping your camera off and your mic on mute if you're not speaking, this is to reduce possible activating motions and sounds. We've all been on Zoom calls where there's somebody on camera way down here in the corner who's doing something that's very distracting. So if you're just here to observe, welcome. Thank you so much for being here. But if you can go ahead and keep your camera off and your mic on mute, please put questions in the chat. One of our board members, Michelle, is here monitoring the chat for questions, and also alert us to any issues that might come up. Everybody who is a presenter here, including myself, either has misophonia or is very familiar with it, and we will be doing our best to reduce or just eliminate any kind of triggering motions, triggering sounds, but we're not perfect. So if anything comes up that's really distracting, please alert us in chat. Michelle will jump in and say, hey, can you stop doing that thing? Totally understandable. We welcome that. Also, feel free to use the automatic transcription button. In Google Meet, there's a button below the window that has a little C and C for closed captioning. If you click that, it will turn on closed captioning for you, and you can read what we're saying as an automatic transcription. You can put us on mute and read this session, if that helps you at all. It's a feature that we really like. However, with great power comes great responsibility. You also all have access to the Present Now button, which is a square with an up arrow. If you click on that, it will share your screen. Please avoid doing that accidentally if you can. That's so Dean can share his screen here in just a moment. And one other thing I do need to mention is that the nature of the topics we're going to be covering, we're actually going to be covering 3 or possibly 4 different papers / projects / studies that our guests have done. As a part of that, we will be discussing and mentioning specific misophonia triggers. We won't be playing them, you won't be hearing them, but there is kind of an anecdotal understanding that some people just hearing them mentioned can be triggered. I just want to give you a heads up that the nature of these studies, we have to talk about specific triggers, but we won't be playing them. So please let us know in the chat if there's anything that is troublesome with that. Lastly, this event is being recorded. So you are all being recorded, and we will be posting the video hopefully this evening, if not tomorrow, and I'll send out an email. It'll be on our YouTube channel and our website for free to watch later if there's something that is compelling, or if you want to share it with somebody, please do so. Like I said, my name's Chris. I'm the founder of soQuiet. We're a nonprofit dedicated to misophonia advocacy and support. Since you all are here, you're interested in research, I want to tell you about a few of our research initiatives, and then we'll introduce our guests. First of all, misophonia student research grants. We provide up to $1,200 to graduate students who are researching misophonia, misokinesia related conditions. One of the studies we're going to be talking about today is one of those fortunate grants. We love giving out money for misophonia research. So if you know anybody who is a grad student who would like to study it, have them contact us. We also have the Misophonia Research Pool, which is the only centralized misophonia research participant registry that we're aware of in the world. What does that mean? Well, a common question is, how do I participate in misophonia research? And until now, there really wasn't a clear, concise answer for that question. The misophonia research pool is free for everybody. You just sign up, and any studies that are happening where they could use participants who experience misophonia or similar conditions, we put them in touch with you. And lastly, the soQuiet science sessions. Sorry for all the S sounds, but that's what we're doing here. This is our attempt to bridge the gap between the general misophonia community and the academic research community. There's a lot of really amazing work happening in the research world on misophonia from many different aspects, but that doesn't always make its way to the people who have misophonia or are affected by it. And these sessions are our kind of informal way of trying to bridge that gap. And there's some URLs you can get more information on all of those. All of our programs are free thanks to your tax-deductible donations. In the US, we're always happy to get donations at soQuiet.org/donate. You can also text the word soQuiet to 53555 and donate on your phone via text while you're waiting in line at the bank or whatever it is. It only takes about 5 minutes, but everything we do is free for all, open access, thanks to our kind donors and volunteers. If you have any questions, comments, or concerns, please put them in chat. Michelle's monitoring the chat graciously. But if anything else comes up at any time, I answer every email, no matter what it is. You're welcome to contact us at hello@soQuiet.org. We're glad to hear from people in the misophonia community, so feel free to reach out. And now I'd like to introduce our guests. We've got 3 student researchers who are really prolific and producing some pretty amazing and diverse work very recently. So if you all wouldn't mind introducing yourselves, if you want to start, I guess, Danielle, if you want to tell us a little bit about yourself.
Sure. So I'm a master's student at the NSERC EARS Industrial Research Chair in Near Technologies. And I have misophonia myself, and that is what has motivated me to work on my master's project, which is about an in-ear device for misophonia.
Cool. All right. Heather, thank you.
Yeah, my name is Heather Hansen. I am a 6th-year PhD student in cognitive psychology at The Ohio State University, and I have been studying misophonia in a variety of different ways, from what sounds tend to trigger to what it looks like in the brain, that type of stuff. I was fortunate to meet Danielle at the Misophonia Convention last year, and a collaboration was born, so we're excited to tell you about it.
Cool. Thank you. And Dean?
Yeah, hello everyone. My name's Dean. I'm an undergraduate neuroscience student here at Ohio State. In the past few months, I've been working under Danielle and Heather in a neuro lab studying misophonia. I don't have it myself, but I've been involved in their research, and it's been pretty interesting. So I'm excited to talk about that today.
Awesome. Thank you so much. Okay, we've got a lot to cover. This team has been incredibly prolific. There's actually 3 very relevant, very important projects / papers that they have all produced and released in just the last month, 2 of them just in the last week, 1 in the last 48 hours. So that's the one we're actually going to be starting with. It's called the Free Open-Access Misophonia Stimuli Bank. And this was funded by a misophonia student research grant, which we talked about earlier from soQuiet. And I believe Dean is going to tell us a little bit about this project. It's kind of fascinating. So I'm going to hand it over to you, Dean, if that's okay.
Yeah, thank you. All right, I'm going to attempt to share my screen really quickly before I get started here. Okay, can everyone see this?
Yes.
All right, perfect. Yep. Okay, so yeah, as Chris was saying, my name is Dean. I've been working on this FOAMS project, which is a free open access misophonia stimuli database. So this is part of the research that we've been doing for the past couple months. So I'm going to talk about kind of what this database looks like, and kind of the methodologies and the reasons behind creating it. Right. So to start out with, misophonia, as we all kind of know, right, it's incredibly diverse and it affects everyone differently, right? There's a wide variety of triggers out there in the literature. And so studying it has kind of proven to be a really complex question, right? This idea of how do we study misophonia? This has many answers, but in short, right, the obvious one would be sounds, right? After all, we have sort of a consensus definition of this disorder already, right? It's categorized as a decreased tolerance to specific sounds or stimuli associated with such sounds, right? This is something that we know to be true. We understand misophonia for what it is. However, that doesn't necessarily make misophonia research any easier, because although we know that sounds can elicit responses in people with misophonia, the next question becomes, all right, which sounds should we study, right? Really quickly, I believe as people were joining this room today, Heather put something in the chat. So if you're comfortable, you can go ahead to that poll and share with us some of your personal misophonia triggers. And I believe here, before we move on, I think we have a way to visualize that. Heather, if you want to take over the screen share for just a second, and we can take a look at what we're doing here. Let me stop presenting really quickly. There we go. Okay, cool. Yeah. So this is in live time. These are what everyone today has shared so far. So you'll notice that bigger words correspond to more instances of people saying them, right? So just some common triggers. Looks like chewing is a really common one. Things like snoring, gum chewing, crunching. Okay, this is interesting. It's interesting to note where these sounds are coming from too. You'll notice a lot of these seem to like associate with mouth sounds, right? Theo, I believe the poll's in the chat there. You can also scan this QR code to have like some direct access here, and you can share with us anything. So you'll notice that there are lots of mouth sounds here, right? Oh yeah, really quickly before we get started, I know Chris touched on this a little bit earlier. We will be discussing some of these triggering sounds throughout the rest of the presentation here. So if at any point anyone needs to step away or anything, it's completely fine. I'm more than welcome to, right? But yeah, so if you'll take a look at some of these, some of them around the peripheral too, they don't necessarily have to do with mouth sounds, right? I see water dripping here, oven clicking, that's a good one. We call those like mechanical sounds in some of our research, right? Humming, whistling, some more mouth sounds too. Conversations, yeah, just talking in general, right? Tapping, big one. Yeah, so we can discuss these a little bit more in this. Popping, yeah, that's right. So these are common triggers that we have seen like in the literature already, right? So I'm going to go ahead and grab this screen share once more. Okay. Yeah, so as you can see, there is a wide variety of triggers that are reported, right? And these triggers can span a variety of categories as well. So as I mentioned, mouth and nose sounds. These are just some triggers that we found from the existing literature already. Mouth and nose sounds, oral nasal sounds in our study, we call them. These tend to make up the majority of sounds that elicit a response in people with misophonia, right? Things like chewing, those big words that we tended to see just in that graphic, right? For all different categories, things that come from hands and things like clicking, tapping, like things rubbing together, those sort of sounds that can also be triggering. Leaf blowers, things like mechanical sounds as well, things that aren't necessarily associated with humans, right? So sometimes like object sounds. We saw the stove clicking, the clock ticking, but also sometimes animal sounds as well that just aren't humans, right? Dogs barking, those sort of repeated stimuli that can be triggering for some people. So this is a broad list of triggers, right? So then the follow-up question for researchers then becomes, okay, we know that triggers exist. How do we acquire these sounds, right? As researchers, what do we do with them? As you can maybe imagine, there are kind of a lot of logistical barriers to misophonia research, right? In studies in the past, it has been very time and labor intensive to study misophonia, right? Whether or not we're our own sounds, we're recording our own sounds, or we're just compiling a list of triggers, there are lots of different variables that make it pretty difficult to consistently and accurately study misophonia, right? Studies in the past have used triggers, but they sometimes vary, right, in the content of what the triggers actually are, in the length that they're exposed to subjects, right, or even the number of triggers used sometimes. That can vary from study to study, right? There can also be individual variations within triggers that cause them to either be triggering or not for certain people with misophonia. I'll touch on this point later. This variation is kind of an important aspect of the sound bank that we've created, right? Just for some context, this variation is rampant, right? It's all over the place. These two sounds, these visual representations of some sounds that we would work with, right? These two are actually coming from the same sound. They're both water drops. However, you probably guess from what they look like that they differ substantially, right? This one up top is pretty clustered together. Maybe the amplitude that it displays right here, it doesn't peak. It doesn't have super high peaks or super low valleys when compared to the one beneath it, right? Which is a lot more punctuated. Maybe those are single drops of water falling. We're not sure. There are these periods of relative silence or just quiet noise between them, some activity going on towards the end there. So this is kind of an example of the problem that we run into, right? These would both, in previous misophonia studies, be categorized as water drops. These could be the same trigger, even though they vary substantially, right? So then the research question that we are attempting to face is, okay, how can we reconcile this, right? How can we accurately and consistently study misophonia? And our kind of proposed solution lies in this creation of a sound bank. This idea that we are going to gather misophonia triggers into a freely available online and open access database for anyone to use, right? Collaboration is a big aspect of what we're going for here. And the idea here is that we would arrange misophonic triggers in as detailed of a way as possible. We'd compile them, we'd label them, and we'd further even categorize them based on where they're coming from. We'd also describe the sounds that are going on in them, right? So if you look on the right-hand side here, these are actually all of the labels that we ended up working with in this sound base, or in this database, that is. So you can see that we further divided them into what we call their parent sounds. So they're oral nasal sounds, right? Hands and feet, they're like non-human sounds as well. And further, within each one of these sounds, we have actually multiple instances of them too, right? So exhaling, right, is one of the sounds of oral, for example. We have like 5 examples of exhaling. Some that are like fast, like punctuated breaths in and out quickly, right? And some that are slow and deeper. So there would be multiple examples of the same trigger, and we would describe those variations that take place within those, right? So all of this in an effort to really specify what the triggers are, the researchers are able to use. And then further, along with like the categorization idea, each one of these sounds has a specific ID number, and we have spelled out our methodologies for like how we end up actually labeling the sounds, right? So everything is entirely traceable, and there's an emphasis on that idea of reproducibility, right? If this database is going to be open to all different kinds of researchers, we want everyone to be able to replicate studies, right? That's just good for science, right? Being able to share those types of resources. So yeah, the overarching goal here is making something well-defined, that's specific as it can, but is also completely available to anyone who wants to utilize these triggers, right? Now, I realize that this is kind of an abstract idea to work with, this whole like kind of sound bank. So I do want to take a second and kind of walk through what it takes to actually create one of these labels on the right-hand side here. So on my end, step 1 looks like gathering a sound and visualizing it, right? So we use stock sounds that have been uploaded to freesound.org, just where users can upload different instances of sounds from everyday life, right? And we compiled a list of historically common misophonic triggers. And so step 1 for me would be taking a sound like this and visualizing it just through some software. Yep, there's the freesound.org link. So let's say, for example, that this sound here is chewing gum. And I don't know anything about it yet. I just know that I can tell. And at some point when I listen to these 10 seconds, there should be an instance of chewing gum in there, right? But now my job is to label this and describe it as best we can, right? So step 2 is just that. We go ahead and start labeling. So I listen to the whole sound, and I'm searching, I'm listening and thinking about where chewing gum actually takes place, right? So in this particular one, it looks like chewing gum happened for all 10 seconds for the entire duration of the sound. So down here, next to this arrow over here, this first long, thin blue line here, I would highlight where chewing gum is actually happening in this sound, and then I would label it. And it just so happens that around the 6.5 second mark, there's another sound that came up in this file, right? And that was talking. So I heard somebody talking in the background of the sound at the same time that someone was chewing gum. So I add another label here, and this C2 label lets me know, this C2 here is actually, it's a salience label. So it lets us know talking is going on in the foreground or the background. This talking is in the background, so we call it a C2, as opposed to the chewing gum, which happened in the foreground. It's the very first thing that like I would hear. It's very much like present. So it's a C1, and the talking is a C2. So for this particular sound, it contains 2 labels. That brings me to step 3, which is taking all of these labels and creating a taxonomy using them, right? So for chewing gum and talking, the idea here is that we would keep an ongoing list of all of the labels that we have, right? And chewing gum, we would, in this taxonomy, we would describe the label itself, and we'd also make note of the parent sound. If you think back to that sort of branch visualization that I had a couple slides earlier, this is where we're working right now, right? You'll notice that talking and chewing gum are both underneath this parent category of oral nasal. So just in this one taxonomy and in these labels, we now have information on the duration of the sound, the identity of the sound, the salience of the sound, whether or not, like essentially the volume at which it takes place, right? Its parent sound, and then also where that parent sound is originating. If you'll remember, this oral nasal connects back to a human node, right? So for chewing gum, that might be a little bit obvious, right? Just because few things besides humans chew gum. But for some other stimuli, things like exhaling, it's useful to know the parent categorizations too. Oh, and then really quickly as well, having a sound that has multiple labels in it could actually also be useful when conducting research, because if we have a sound that contains salience differences, right, one in the very foreground and then someone talking in the background, this might be useful if a researcher, say, wants to isolate just chewing gum, or want to look at how misophonic triggers can occur in sort of like ambient environmental noise. So they now have, yeah, it could be a double trigger, right? They could now be given the option to like see how a background noise could impact misophonic responses, right? Or they could just choose to use the isolated sound. So we're pretty excited about this database because there are tons of different future directions that it could take, right? In kind of the immediate sense, this helps a lot with the standardization issue that I talked about earlier. Those logistical barriers to studying misophonia can help kind of be smoothed over if we have a bank of standardized misophonic triggers, right? This allows for researchers to use consistently like the same types of stimuli. And then this goes hand in hand with the idea of resource sharing, right? Studies have follow-up studies use these same exact triggers, or they can just be overly like more specific than they have been in the past, right? And this is good for the scientific community. It helps create more consistent findings, right? Things that are able to then be tested further and with a higher degree of accuracy. The variation in our sounds is also a pretty important aspect. And we kind of have 2 types of variation, right? The first being the idea that we have variation among different triggers. So in the past, we've had some comments from participants in misophonia studies saying, oh, well, in this particular one, my triggers weren't really well represented, right? Maybe they just didn't contain, the study did not contain sounds that elicited a response from them. And our database hopefully can help solve that problem because we have such a breadth of wide triggers to begin with, right? And they span multiple different categories as well. And further, although we have a ton of different like categories of sounds, we also have variations within those sounds, those 5 different examples that I was talking about earlier. Another participant at one point said that, like, oh, well, in this particular study, for one reason or another, the sounds just didn't quite hit for me, right? I was maybe reacting to something that I shouldn't have, or that I know I don't react to in the past, or vice versa, right? With our variations in these 5 different examples of each type of label, the idea is that we're increasing the likelihood that there is at least some instance where somebody would elicit a response, right? Say if you know that you respond to pens clicking or something, but you don't really respond too well to pens clicking slowly. The idea here is that we'd have both fast pen clicking and slow pen clicking. So we probably cover more bases from an experimental standpoint in that sense. There are also a ton of different directions and implications that this database can have across different fields in neuroscience, right? The first being psychology and neuroimaging. We can start to answer some questions that we haven't really been able to approach consistently before. Now that we have a free, like, open database of consistently labeled sounds, right? So things like, what happens in the brain when you specifically are exposed to your triggers? Because we have such variation that's also standardized, we can really take a look at what elicits a response in test subjects, and then take a look at their brains doing neuroimaging studies and see what misophonia actually looks like and what responses look like from a physiological standpoint, right? There are also implications in the fields of acoustics and hearing technology. This is because we use computers to analyze these sounds, right? And the fact that we use computers means that we can also start to answer some questions that humans couldn't really approach to begin with. So the computers can visualize these sounds physically, right? So we can take a look at questions like, do trigger sounds tend to be a certain pitch? Humans might not be able to pick up on these sort of regularities within trigger sounds. But if we visualize them using computers, computers can go in and actually pick the specific, the key tones in the sounds, or things like that maybe are common among different misophonic triggers. And then further, can machines then learn to detect these sounds, right? These are questions that we couldn't really answer without having standardized sounds and without using computers to aid in them too. So my colleague Danielle, she's actually been doing some of her own research into the hearing technology aspect of this. Danielle, do you want to speak a little bit about that?
Yeah, sure. So the device that I've been working on developing in my master's project, in contrast to traditional earplugs or noise-canceling headphones, which block out all sounds all the time, what I'm really aiming to do is to have a device that can record the sounds around you and pass them through if they're not triggering. So really only blocking out sounds when the wearer is triggered. And one real challenge of developing a device for misophonia is that people are triggered by very different specific sounds, as we saw in the poll. And so we need to have data on these sounds, and a variety of data in order to have algorithms that can recognize these sounds to know when the wearer is likely to be triggered or not. And so I hope that by providing the raw data, providing the labels at every step of our annotation process, that in creating this database, we also offer the flexibility that people from different fields of misophonia research, that they can reuse these for different types of research questions. So, Chris, I think now might be a good time to pause for the Q&A before we move on to Heather's presentation.
Sure. Yeah, thank you, everybody. That was fascinating. And part of my role here in the science sessions is to play the layperson. I have no background in science or medicine. I barely passed algebra in college. So, yeah, these two projects, we posted links in the chat, but to kind of summarize, and then we'll open it up for some questions about those specifically before we move on to Heather's neuroimaging study. If I understand correctly, these are, well, the FOAMS database is meant to be a standardized set. And one of the things that was kind of new to me is that what tools you use in your research affects the outcomes. We talked about this with Dr. Gregory and Dr. Vitorto on a previous science session. Right now, there's about a dozen different misophonia assessment questionnaires out there in the world, and which one a particular researcher decides to use can influence the outcome of the study. And extrapolating that to this, if somebody's going to research misophonia, they need to trigger people, unfortunately, and so they have to collect their own sounds, and those sounds can be all over the map. There's no consistency to the tools that are being used. And if I understand correctly, this is an attempt to have a free, consistent tool that future researchers can use. Is that it? Am I summarizing that correctly?
Yeah, I think you did a great job there, Chris. And you're hitting on a good point, because you might see research come out where someone says, hey, I found a brain region, or I found a response to this, chewing sound in particular. And someone else might come out and say, well, like, no, I don't find that same chewing response. And we don't know whether or not there's something actually interesting about, like, the differences in those, maybe one is more higher pitch or the other, whatnot. It might just be due to the fact that you use a slightly different version of the sound. And so our kind of goal is to increase the replicability of misophonia research, because now all researchers could have access to similar stimuli, and we'd be able to better kind of tease apart what nuance actually exists.
Yeah, it's got a consistent foundation. That's pretty awesome. And we're glad that it's just been released in the last 48 hours. So this is a brand new tool and research on that tool. And to sort of summarize Danielle's paper that is not even a week old, I don't think, as far as being published, she was taking, and her team were taking, an open source platform, software and hardware that is normally used for prototyping hearing aids, if I'm understanding correctly. There's one right there. This is a freely available thing that people can use to develop new hearing aid technology. Her team is hacking it, for lack of a better term, and trying to use it for misophonia and other sound intolerances, if I'm understanding correctly. And one of the things that we see mentioned every now and then is the dream of having programmable noise-canceling headphones, where somebody could program their specific triggers in real time, and the headphones would know that those are triggers and specifically filter those out, but let other sounds through that aren't triggering. Is that kind of what you're shooting for?
That is pretty much exactly what I'm shooting for. Yes. Although I will say one of the there have been roadblocks in the process. Like it works very quickly, and it can run conventional hearing aid algorithms. We can play masking noise on it. You can pass the sounds through. You can filter them. But having algorithms like detecting whether the wearer is stressed using physiological signals, or detecting whether a trigger is present in the outer microphone data, these require a lot of computational resources, and can also take more time. And so what we did in my project is we used a separate a PC, like an external computer, to run those higher resource algorithms, and then used built a processing pipeline that we made open source on GitHub to use the decisions made by a detection algorithm to change the audio processing happening on this on the TIMPAN, on the open source hearing aid.
Cool. Any questions? Do we have questions in the chat, or does anybody have any questions about these two projects specifically?
I have a question.
Okay, go ahead.
Thank you. This is Cheryl Carter, and I'm wondering if it wouldn't be useful during these research studies if you asked participants to BYOT, bring our own triggers. I mean, we're BYOB into the party, why not, right? I think then you'll definitely elicit the responses that we as misophonias suffer through. And then my second question is, have your research studies determined any correlation between hearing sensitivities and other sensitivities? Like, I've been labeled as a highly sensitive person, so my emotions are at my surface. I have a very active smell, touch. I mean, all of my senses seem to produce some issues. So I'm just wondering if there's any correlation in these studies or any future studies. And those are my two questions. And thank you so much for the work that you're doing.
I think it's a great question. Actually, a great couple of questions. Anybody want to field that?
Yeah, I think I can speak to the first one. I'm not sure how well versed I am on the sensitivity issues, Danielle and Heather. But yeah, so in terms of participants their own trigger study, I think that is a great idea, right? I mean, that's kind of at sort of the core of what we're trying to do here. As Chris was saying, it's an ugly truth, but a lot of misophonia research is figuring out the best, most efficient ways to trigger people, right? And so including participant sort of inclusion there, right, with like them bringing sounds that they know to be triggering to them personally, I'm sure that would greatly improve the precision of these studies, right, and the outcomes. In terms of how that applies to the sound bank that we've created, I could see researchers using the variations in sounds that we have here to maybe before a study is actually conducted, maybe they have a group of people who know that they respond particularly well to lip smacking or something like that, for example. So then researchers could give them those 5 instances of lip smacking that we have in our database, and then record which ones out of those 5 they respond to the most, or that elicit, you know, a strong response or a weak one, whichever one they're looking for, right? And so in that sense, they could sort of bring their own knowledge of what triggers them best to that study, but then also keep it consistent with using sounds that are repeated throughout studies, right? That's interesting. I feel like there are a lot of implications there with being your own triggers. It's a cool idea.
Gotcha. Thanks, Dean. Anybody else have something to add to that? And then we'll move on to Mary Rachel's question and then the next study we're going to be covering. No? My understanding, there's, again, I'm not a researcher, but if you keep track of what's going on in other communities, like the autism community, the ADHD community, the highly sensitive person communities, there's a lot of interest in understanding that part of the question, which is our senses are really mysterious in a lot of ways, and what people are sensitive to and why they're sensitive to them across different senses, whether it's interoception or olfactory or whatever. There's a lot of interest in learning a lot more about that. We just don't know a whole lot at this point, and hopefully we will soon. That's my understanding. Okay, we got a couple of quick questions for Danielle about her project. People are really interested in the concept of programmable noise canceling headphones. Andrea asks, would the audio device be customizable with each user's specific sounds? And what would the triggering sounds be replaced with? And prior to that was a question on when is it going to come to market? You've got some potential buyers in the pipeline here, Danielle.
Okay, I guess I'll answer these in the order in which you asked them. Yes, the goal is that it would be customizable with each user's specific sounds. However, because this data, and even our database, you know, the phones database, it's not comprehensive enough to cover every single trigger sound. That's why we would love to hear from you, and even have recordings from you with your own trigger sounds. I personally have not worked on having an algorithm that can detect all these different trigger sounds and be customized to everyone. However, I do think that in your devices, because they can also record physiological signals, that could be one avenue in which we can customize a device to each individual based on their stress response. And the second question, what would the triggering sounds be replaced with? Just silence or different sounds? That's a great question. I think there needs to be a bit more research on the utility of masking for misophonia. And I would hope that using, an open source tool rather than just like an in-house a device developed by a lab, if we could have more studies on how people report that this specific masking noise helps them or does not, we would know more about what's the most effective way to replace trigger sounds. So and the hitting the market question, I honestly I don't know, but I think slowly we're getting there. Like you are seeing more and more products that have like a transparency mode that where you can easily toggle between blocking out all sounds and letting them in. We're seeing I think New Hera, they have a device that's actually marketed for sound sensitivities because they specifically try to filter out all non-speech sounds. So I think slowly we are getting there, and we just have to try to make all the resources for misophonia specific development available, and hope that that these efforts can get more funding. Yeah.
Thanks, Danielle. All right, before we move on to the next study, Mary Rachel's had her hand up for a bit. Do you have a question? No? Well, if you come back, Mary Rachel, we're happy to answer it. All right.
Can you hear me?
Oh, go ahead. Yep, you have a
Oh,
question?
so sorry. Of course, I have my sound off. I was fighting with my daughter. Thank you, all of you, so much for what you're doing and for being here. I cannot tell you how appreciative I am, and I know as well as the other people that are here, and Chris, for conducting this. The thing is, and here's how I feel with the research, right? You can try to replicate the sounds. There is a difference between the authenticity and the place and the time. So I feel it would be really important to get out there. If you guys, you have a team and you can get out there and actually follow people that actually have, right? Like a reality show. I already wrote it down. I'm a casting director and producer. I already have a reality show called, it's now Trigger Me. It's the title, Trigger Me. And we're highlighting all of you beautiful people, researchers, following these misophonia sufferers, going da, 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 and following them and then doing the research, implementing. We can talk about that later. I think that there is an actual possibility there, but you can replicate sounds all day long. I can tell when it's fake. It's the situation. Someone brought this up. Sounds can vary. Where are you? What are they doing? For instance, I'm in the Vons supermarket, and there's a lady on her cell phone on speaker, which is also triggering, and then she's crunching every bag of Doritos, crack, 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 picking, crack, 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 feeling up every bag, crunching and going. And I'm like, how do I deal with this? So I secretly, I'm record it. I'm like, oh my God, this woman's she's doing this. These are the things that need to be shown. These are the things that I think will help. Like science is, yes, you can try to replicate and do all these things. But I think with misophonia, because it is so diverse, and listen, who does want to listen to someone clipping their nails in general? Who wants to listen to someone slurping or clicking or sniffing? No one wants to listen to that. It's not a nice sound. But what the difference is, the neurological differences with misophonia is either I'm going to kill them or I'm going to run away. How do I, we tolerate so many sounds on a daily basis. So I feel like the deeper level here is, yes, we can have all these sounds to trigger, great, but we have to be in the real life situation to really get the results. And if there's any way that I can help, or there's a way to do that, because I know that you guys just have this certain amount of money to research, it's just an option. I just love what you're doing, but I'm like, I have so many ideas on how, you got to follow people in real life. I feel, really, and I don't know if that's optional to you.
Good question. Good points. Do any of y'all want to field that real quick? We do know that context and source can be a factor.
I think a lot of that really resonated with me, and that's part of the reason why we took these realistic field recordings from Freesound, and why we made this Google form available, so that if anyone from the community feels that their triggers haven't been represented so far, that they can actually record real triggers, upload them to Freesound, and send us a link, and we'll make those links available to researchers. Yeah, I completely agree with you, and that also that's why we have included even stimuli with multiple labels, because we noticed that can make the situation more that can make the stimuli more realistic. You know, like people, they don't just swallow in isolation. You know, maybe they're chewing and then they swallow, and then they I don't want to make the sound, but you know, make the sound after people
make the sound. I'm ready. Trigger me all the way. Like, just trigger, like let's get it done. Like, we need to see what the reaction is. People need to see how we feel. I think that needs to be visually recorded, because I struggle with my daughter. I'm a single mom, and she's triggering me on purpose right now, and it's hard, and I'm struggling. And this is why I'm sitting on this session, because I don't know if I can take it anymore. And it's fight or flight. And most people are like, well, I just run away. Well, I'm fighting more now. Right. And I don't know if anyone else is experiencing that. And how do we know that there haven't been people in the past that haven't done anything really outrageous because of this condition? And it really is. And I know there's sort of, and it's gotten way worse as I've gotten older during the pandemic. You know, it sort of started when I was 12, and I'm like, someone's eating at the table, blah, 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 tolerate it. But this past 2 years, it's like, it's affected my entire being. And new sounds happen every day. And it's very hard. And I can do all the work I can do. I can take therapy and meditate and work out and all these things. But if someone's triggering me on purpose, for me, it's going to be a rough ending. And it's really hard. It's really hard. And so I just want to thank you. I'm very appreciative of you all being here. And I want to thank you again. I'm enthusiastic about our very creative ideas, and I know you can probably hear that, and I know, Danielle, you suffer. Dean, I couldn't hear. Do you also have misophonia?
No, I don't regularly.
Lucky you. Isn't it fun being around people that do have it?
All right. Thank you so much. Yeah, I think we can all relate to your experiences and hopes for where research can go. That's actually, Mary, a nice segue into the last paper we're going to cover, which was published about a month ago, I think. And Heather's sharing her page. We'll go back a little bit. Let's step back a couple of years as a lead-in, if you don't mind, Heather, to the Kumar studies, which are very well known. They were very widely covered in the media. People outside of the misophonia research community probably came across the Kumar studies. And the last one from 2021 was called The Motor Basis for Misophonia, and it was widely reported as finding the cause of misophonia, which it didn't. But it gave us some interesting new information and some things to explore. So I'm going to go ahead and hand it over to you, Heather, because there were a few things from that study that I felt were the next logical questions for us to study, and this paper is doing that. So I'll go ahead and hand it to you.
Thanks, Chris. Yeah, kind of shifting gears a little bit from the Sound Bank project. But as Chris was mentioning, there was a previous study that was done that showed some connections in the brain between areas of your brain that process things like mouth movement with areas of the brain that do things like auditory or sound listening. And as Chris was mentioning, this rocked the mesosphere. But one of the things that I was really curious about, and kind of circling back to our very first couple slides here, people are triggered by a lot of things that are more than just mouth related. So I credit their study a lot for really motivating some of the work that I'm going to show you today, which is looking into whether there's actually neural evidence in the brain for not orofacial or not mouth face kind of triggers in misophonia. So what I'm going to show you is a kind of nuanced scientific neuroscience article And I recognize some of you might already be up to speed with some of those terms, but for those of you who aren't, let's bring everyone up to speed. So we're going to do a quick neuroscience crash course in a couple slides. So just to explain some of the main kind of players you need to know about in this research, here is a diagram of the left half of your brain. You can see the little eyeball here, and you might notice by the different colors, your brain can be subdivided into a variety of different sections that each kind of do their own thing. One of the kind of sections I want to focus on here is this section here highlighted in orange, which we call the motor cortex. This is a piece of your brain that controls and is in charge of movement of your body. And as you might notice on this really quirky diagram here, there are special sections on this strip for each part of your body. So for instance, this part here is going to control movement of your face, this part movement of your hand, etc. So it was this piece of cortex that was kind of the main spot they looked at in the previous paper, looking at, okay, movement of the mouth might have some role in misophonia. But besides this motor cortex, I also want to draw your attention to this spot I have highlighted here in blue. This is a spot in your temporal lobe. It's kind of right by your temples or right behind your ear. And this is auditory cortex. This type of cortex does a lot of different things, anything from, lower level, just inputting the frequency of what sounds you're hearing and kind of associating what the different sounds are, but also higher level, figuring out the content of the sound or figuring out if it's speech, what the words are, what the person is saying, that kind of thing. So all things kind of hearing related. And the last part that's relevant for this research is what's called the insula, which isn't actually on this diagram because you kind of have to open the brain a little bit to see it on the inside. But it's this piece here in green, and it does a wide variety of things. Of note, things like the subjective response to pain. So what you find painful versus what someone else might find painful. It does things like processing and dealing with emotions. In particular, disgust has been really commonly associated with the insula. And lastly, it does a lot of attention or alerting processes. So focusing your attention on things that you need to care about and kind of ignoring irrelevant, distracting things. So hopefully you can see just based off, these parts I've highlighted that there might be some relevancy to misophonia in these areas of the brain. So what can we do now that we know areas of the brain do different things? I'm going to introduce you to two methods real quick that are relevant to my work. Both of these methods are things that we get from a MRI scan. So if you ever had a magnetic resonance imaging scan, you've been put into this giant metal donut. If you ever wondered what kind of things we can look at in your brain, let me show you a few. One thing is called a functional MRI image, or fMRI you might commonly see. And this is a way of asking the question, where in the brain or what brain regions are active when you do something? So for instance, if I had you lie in this scanner and I showed you videos of puppies or something, I could then see what part of your brain really lit up when I showed you those videos, and then kind of deduce where, puppy regions might be or where visual input might go or things like that. So I can take something like a task to determine where in your brain is a region that processes that. And once I have things like that, once I know where in your brain is the puppy region or the visual region or whatever, I can then figure out how those regions connect to one another. So one of the other things I do in my paper is what we call functional connectivity, which is basically asking the question, which brain regions talk to each other or are in sync with one another when your brain is doing something or not doing something? So for instance, in this diagram here, I can show you a couple different regions, A, B, or C. And what we can do is just measure the fluctuation of these regions just across time and look at how closely those fluctuations align. So for instance, the yellow and orange regions kind of tend to go up and down in the same way. Those regions, we would call them connected. They're in sync more. Compared to the blue region, that's pretty off, right? That is not connected to B or C as well. And what you might see is kind of a pictorial way of representing that connectivity. This is what we call a matrix of values, where we can take all of the regions we care about, A, B, or C, on the vertical as well as the horizontal, and compare, okay, how much does A connect to A? And we can get a value here. So for instance, I'm showing you those are more connected to each other. Oops. Or B to A, you can see is less connected to each other, etc. So we can basically measure connectivity from every brain region we care about to every other brain region we care about. Now, the way that I did this in my task is for fMRI, I was really curious about exploring this question of oral or nasal aversion versus something that was not oral or nasal. So the quick way that I did this was giving participants a task where they either had to move their mouth in the scanner, so they're physically saying these phonemes out loud. So they're saying ba, ga, ra, da over and over, moving their lips, moving their tongue, things like that. And then I also had them physically tap their fingers in the scanner. So I'm getting movement of your fingers. And in essence, I'm trying to get at this motor cortex I showed you earlier, finding in each individual person where their mouth face region is versus where their finger region is. And in fact, when I show you data from my own study, I can then kind of define this region that really activated when they move their mouth. I can call that orofacial cortex, or an orofacial part of the brain. And the part of the brain that's really active when they tap their fingers, I can call finger cortex in their brain. And if I do this for each person individually, I get a really precise estimate for each person where their region is, which is a great thing to do with neuroscience research. And then lastly, now that I have those regions personally defined in each person, I can take the different things I care about in the brain. So in this particular case, orofacial and finger cortex, and the two things I showed you earlier, auditory cortex that does things hearing, and insula that does disgust or alerting attention. And I can measure connectivity but in and between those regions. And I can do that for each person in my study. Each person's going to have slightly different connectivity between those regions. And they're also going to have slightly different misophonia levels. So I can use that to my advantage. I can subdivide my group based off what their misophonia levels are. So in my particular study, I split them up into those with higher misophonia and those with lower misophonia. And now I can analyze and look at what connectivity differences kind of group them. What is different between those two groups? It is this that I'm going to show you as my results on this next slide. So just to orient you quickly to this graph, I'm going to show you how connected these regions are with the orofacial or mouth region on the left graph and the finger cortex or finger region on the right graph. And I'm showing you connectivity. So lower down on the y-axis, we have those areas aren't connected at all versus upper on the y-axis, those are really strongly in sync with each other. And I'm doing this both for the two regions, auditory and insula, that I care about. So as prior work was shown, there's commonly misophonia is associated with chewing mouth kind of noises, right? So if I look at connectivity between individuals with higher misophonia and lower misophonia in the orofacial region, I actually don't find differences in connectivity between these regions. And this might be due to a variety of different factors. Maybe the groups that I have with lower scores also just kind of hate gum chewing. Maybe that's more of an aversive noise to both of them. And maybe my study wasn't designed enough to tease those differences apart. However, one of the things that I do want to draw your attention to is the fact that I can look at connectivity with finger as well. And just kind of taking a step back conceptually, if misophonia had nothing to do with finger or unrelated mouth noises, if misophonia was just a mouth condition, there would be no reason to see any sort of differences with a finger region. That wouldn't track. But when I look at the same results from the finger region, I actually do see actual connectivity differences with the insula. So this was statistically significant where individuals with higher misophonia had a lot higher connectivity between the finger region and the insula than individuals with lower misophonia. So what this is telling me is there's a stronger representation between this kind of disgust or alerting region with finger regions in those individuals. And I don't mean to make this like comment of like, finger is the way to go. But what I want to take away here is that we have evidence now that there are non-orofacial regions that show differential brain connectivity in misophonia. So this kind of just gives the general idea that we need to expand our mindset past just oral or nasal noises and kind of look at a broader whole, what else is going on. And I know we've hit 3 o'clock, so I'm going to wrap up, and that's the end of my presentation. I'm happy to take any questions. I know this is a lot.
Thank you so much, Heather. And we can go over a few minutes if people don't mind sticking around for a few minutes, because this is fascinating work. And I read through the paper as a layperson, having read the Kumar study. And if I can sum it up, you're not only replicating the Kumar study, which is very important in science. A single study can only tell us so much. Other teams and other scientists and researchers have to replicate it to confirm that what is being found in a particular paper is correct. And I too is sort of a little not bothered, but I'm kind of bugged that the Kumar study only focused on orofacial triggers, whereas we know most people with misophonia have lots of triggers that are not associated with the nose or mouth. And my obvious question as just a casual observer was, what about those things? And you're attempting to expand on that in your replication of the study, if I understand correctly, by including the finger tapping part.
Yes.
Amazing stuff. And another thing that kind of caught my attention is some parts of the Kumar study you confirmed were correct, but you have reason to kind of cast some doubt on the motor neuron if part of their assessments. Can you explain that a little bit? And then we'll get to some questions in the chat.
Yeah, so that study focused on the fact that you have neurons in your brain that hypothetically will activate both when you are doing something and when someone else is doing that same thing. We call those mirror neurons, right? So just some overall knowledge. Neurons themselves are super tiny, and they're really hard to actually, in fact, you can't really study them in something like an MRI. MRI is not fine-tuned enough to look at individual neurons. So one of the just kind of general takeaways is that if you are only using MRI as a method, you can't make a definitive claim about what a single neuron is doing. So both in that that theory would need to be validated using actual electrical recording for that hypothesis to be stated as true. But also, in addition, what I didn't really highlight here is we expanded our search past just motor, but also to sensory motor as well, which I haven't shown here. But we found differences in the sensory part of the brain as well, things that sense what parts of your body are doing. And so the fact that we're showing differences in sensory as well as motor kind of also reduces the likelihood that motor is the sole driving cause of misophonia. It's very technical. Happy to talk more, but hopefully that helps.
Yeah, one of the things that I've learned is that the more we find out about misophonia, the more questions come up. It becomes infinitely more complex the more we look into it. And just a hatred of sound, that's way in the past. That's not even relevant anymore. It's a complicated disorder. And we'll go to a couple of quick questions. The other thing that struck me was the Kumar paper ends with sort of a speculation on acquiring sounds, and your paper intentionally tested that there actually is neurological evidence for other types of triggers. And I'm wondering if the next question might be, a lot of us have triggers that are not associated with body movement at all, like clocks ticking, like Dean mentioned, mechanical sounds. Is that the next thing for us to look into?
Definitely. I think you're hitting a great point, Chris, which is maybe let's expand past just thinking about this as a human thing, or maybe past motor altogether. Obviously, I didn't look at mechanical noises here, but I think that's definitely the next step, to look at maybe what differences in the brain exist when there's a mechanical noise, or maybe playing that in the scanner or that type of thing. But yes, I think broadly, more than mouth is my takeaway here.
Gotcha. Okay, let's go to a couple of questions. We've got some good ones in the chat here. Then we'll summarize. I really enjoyed reading the paper, but then, just learning about what advances are being made is really fascinating. So from Mary Petrie, regarding neural evidence, could this research provide evidence of the neural basis of visual triggers? Great question. Might this be a pathway to equal or even any scientific focus on visual triggers? There have been some studies on misokinesia, but those are few and far between, although interest is growing. So visual triggers, how do these associate with your study?
Wonderful question, and one in which I don't think my current data can directly answer, but I think it'd be very interesting to show participants videos of people triggering. So seeing the visual aspect of a trigger and noticing in the brain maybe there are stronger connections with visual cortex and disgust region or things like that. So there has been less scientific focus on visual triggers. I know that there's maybe one prior neuroimaging work that does show videos that tries to kind of get at this, but that is definitely a step to moving forward to look at.
Awesome. And also Dr. Davydenko's, he was the last science session we did. He's found a weird kind of trick in juxtaposing audio and visual. You can go watch that. I won't explain it now, but it's kind of fascinating with misophonia. Okay, from Sam, did you do MRI comparisons with someone that doesn't have misophonia at all? So kind of a control group.
Great question. The short answer is when you give someone a survey that is a misophonia assessment, chances are this is present in a small extent in most of the population. So what we found is we have people who, and that's why we call it lower scores, is because some people will say like, yes, I'm sort of bothered by gum chewing a little bit. So they'll score like a little bit on your assessment. And it's hard to find someone with zero misophonia altogether, which is kind of my takeaway. So I don't have a true control group here because like you said, I'm kind of just subdividing my scores into higher and lower. But in a way, I think it is difficult to find a true control, I think, in this type of study, is my short answer.
Great question. A couple of more and then we'll wrap up. Do you have any idea what determines if a person will have misophonia? That's the big question. Yeah.
It would be super cool, and one thing that I could do in the future with this type of research, if I am able to find some connection or some brain region that seems to be the driving factor of misophonia, we could potentially scan people before they develop misophonia and be able to tell ahead of time that this person is more likely to get a condition like this. That's way in the future. We have no idea, but it's something that we could potentially look at in the brain and kind of determine.
All right. And lastly, how many misophones did you base these scores on? You had a sample set of, if I'm not
great question.
like 19? Yeah.
Yep. I have 19 participants. This was during pandemic, so getting people was a little bit difficult. But I have 19 people. So you're looking at here, I believe 7 of them qualified as being like clinically or like higher levels of misophonia, and the other 12 are in my control group.
Awesome. Okay, we can go ahead and wrap up. We've gone a few minutes past. I appreciate everybody being patient. We had a lot to cover, and I would encourage anyone to read any one of these studies because there's a lot more information that I found very fascinating, some details on how the studies were conducted and what was discovered. But I want to thank all 3 of you, Dean, Danielle, and Heather, for being here. This means a lot. This is the future of misophonia researchers right here in the room with us, and they're already shaking up the research community with really compelling, fascinating discoveries and projects and resources for other researchers to use as a basis. So any closing words that you all have on what's next before we kind of head out?
I'm just really excited that we were able to collaborate on this, even though we're otherwise working on, kind of different things within misophonia research, and I hope to see more collaboration across fields in the future. Yeah.
Thank you. Yeah, we look forward to more. And Dean, thank you for your presentation. Nice job. If anybody has any other questions after this, feel free to reach out to any of our researchers, or you can email me as well. The kinds of questions you might have based off of these are the things that could lead into the next study. It's really fascinating that some of the things that I thought were a little off in the Kumar study as far as speculation have now been researched as happens to have happened. But thank you all so much. The recording for this will be posted, and I'll also send out an email to everybody with a link to that video and a survey on things we could improve on future science sessions. Thank you all so much. This has been wonderful. I really appreciate you taking some time out of your Sunday to help educate the misosphere on your work. And it's very prolific too.